This is a production of Cornell University.
Initially, when I was organizing the seminar program for the fall, I was very nervous about having a lot of gaps that I wouldn't be able to fill with speakers. And happily, we've filled them all up. In fact, initially, my plan was, if I had to, I would give one seminar on cassava and a second one on maize. And you're really lucky because we filled them up. And I've arranged that the seminar on cassava is going to be a joint or shared seminar between myself and my senior research associate, Peter Hyde, who is going to give you most of the actual data and results from our NEXTGEN cassava project. So I'm going to just give you a little bit of a lead-in and give you some context on the project as a whole. And then Peter is going to show you most of the work on cassava flowering that we've done in pursuit of this project. The NEXTGEN Cassava Project is a large project funded by the Bill and Melinda Gates Foundation and by US or UK aid. And we have, as you can see on this slide, a number of institutions that participate, are participants in this project. The project is led and managed by Cornell University out of international programs. And so Ronnie Kaufman, as the head of IPCALS, is the principal investigator on record. And the lead project is a breeding project for cassava. And I would say that the technical lead is Jean-Luc Janink on that project. So we have these other participants who are on the project doing breeding programs, and mainly our target is in Africa. So we have International Institute of Tropical Agriculture in Nigeria and the National Program for Cassava Breeding, which is located in Umudike, Nigeria. We also have the national programs in Uganda and in Tanzania as participants in this project. And this project has been underway for approximately 5 years, and we've just made the transition from phase 1 to phase 2. And phase 2, as we've sort of gone along, we've picked up contributions from 2 other important places in the world where cassava breeding is taking place. The International Center for Tropical Agriculture in Colombia and Embrapa in Brazil. And there's also even some participation by folks in Hawaii for germplasm transfer from Latin America to Africa. So the project involves quite a lot of participation by groups throughout, and we do our best to become a coordinated project among all the team members. Now, the places that we work correspond to the largest production areas in Africa for cassava. And this map shows you the area harvested in the world. And you can see that Nigeria has a lot of cassava production. In fact, it is the largest producer of cassava in the world. And Africa in general represents about 55% of the total production of cassava. So we have our focus is to work with breeding programs in West Africa, and then these 2 programs, Uganda and Tanzania in East Africa. There's also a lot of cassava production, particularly in Thailand and in Indonesia. Thailand has an excellent breeding program, and they're a bit more advanced. But our work is in coordination with the programs in Africa, where we could really make some progress by improving their programs. Cassava has a number of attributes. So why cassava? One of the reasons is that, in fact, in the world, the amount of production has increased quite a lot in recent years. The trend is that the rate of increase is somewhere between the rate of increase in rice and in maize production, much higher than the rate of increase in production of potatoes and wheat. And so there's been a lot of recognition in recent years about the potential for cassava as being a important crop for the future, and producers are increasing the production. It's a highly productive crop. It grows year-round, so it's particularly suited for tropical conditions where it can bridge between the dry and wet seasons, and it can utilize the land for long periods of time during the year and continue to be productive. It has the reputation for being able to tolerate stresses and in some way be a forgiving crop for farmers. That is to say, in low nutrient or mineral nutrient conditions, it can still grow reasonably well and provide a good production and yield. It can grow in drought-prone soils. And as we go to climates in some parts of the world that are becoming warmer, it is able to tolerate, in fact, it thrives in the warmer temperature that are experienced. So it has potential for becoming even more important in the future. And in that sense, we could say that it's a food security, a good food security crop. It also is producing a starchy root that has many different uses, and so it's very versatile. And so once you have the ability to produce cassava, it can be used for home use and culinary uses at home, various types of products, but it also can be used to provide a product that can be sold for other purposes. For example, a more purified form of starch, tapioca, can be used for a lot of different food manufacturing uses, even industrial uses, as they do in Brazil. Very versatile crop. There are a lot of challenges in cassava production, and that's why we need the NEXTGEN Cassava project. One is that it's not reaching full yield potential. And over the years, it's been recognized as a crop that is has a lot of breeding obstacles, difficulties. One of them is that it is a crop that, in order to obtain sufficient clones that can be tested in uniform yield trials that are replicated at different locations, it takes quite a lot of years to build up those numbers of propagules that are needed in order to do that kind of good trial. And so because of that, the breeding cycle typically takes something like 8 years or more between one set of crossing events and the next one that would be based on phenotypic information collected from the first round. So it's rather been slow to make improvements, and because of that, we feel that there's a lot of untapped potential, and what we need is better methods of breeding that will address be able to make more rapid progress and accelerate the improvement. It has nutritional limitations in that it's purest starch, but it has very little vitamin mineral content. There are efforts that have been successful in introducing higher provitamin A content, and there are other nutritional characteristics that are part of the overall strategy for cassava improvement. And there are it is susceptible to several viral and bacterial diseases that are fairly serious threats, particularly in Africa. For example, in Africa, the cassava mosaic disease, CMD, is a very serious problem, and we have resistance, but it needs to be incorporated into breeding lines. And cassava brown streak is a disease that is expanding from the east towards the west, and currently it is a serious problem in Tanzania and in Uganda. The fear is that it will spread further into West Africa, the largest cassava producing area in the world, and will devastate production if we are not prepared with resistance ahead of that. So that's another of the breeding targets. So the project that Cornell leads is a project that uses a relatively new approach towards breeding, and that's called genomic selection. And genomic selection has the potential to accelerate cassava breeding. It's led by, and this is a diagram from a seminal article written by Hefner, Mark Sorrells and Jean-Luc Janik here at Cornell. And it laid out the advantages of genomic selection over conventional and other breeding approaches. The conventional cassava breeding, as I mentioned a little while ago, involves the clonal propagation of the lines. And in order to have sufficient propagules to do a proper, accurate, multi-location replicated trials, this takes a long time and it slows down the whole advance or improvement process. Genomic selection tries to use a different strategy that will accelerate. And so what we do is we still have the need for high quality phenotyping, but it's done on a subset of the population, a training population. And so this training population is going to be a subset which will represent the breeding population as a whole. And this will be an ongoing process of improving our knowledge of this training set. A training set that in fact involves some changes in the composition of the lines that are in there. What's then coupled with that is that the training lines are genotyped using DNA analysis at markers that span throughout the genome, and it allows the preparation of a statistical model for estimating the breeding value of each individual based on the phenotypic information in the training cycle and the genotypic information from the DNA analysis. So then the program has a model that will be able to predict the potential breeding value of any individual that is produced through recombination and predict it based on the model as opposed to actual field trials of that particular line. What that means is that in the process of line development, after a cross is made and the plants begin to grow, even at the seedling stage, at very young plants, before we can have any estimate of yield, we can take a sampling of the leaves and do the genotypic information marker analysis using DNA methodology, use the model to predict what its breeding value is, and then make decisions on which lines to use as parents for the next round based on that information. It can go much more quickly, and it is actually, with the low cost of sequencing, it can be cheaper actually than doing the phenotyping the old way. So the process then involves accelerating the rate at which this cycle goes between successive crosses and the recombination that's needed and associated with that. And the problem is that cassava has another characteristic that is an obstacle, and that is its poor flowering characteristics. And that's what Peter and I work on. That's what we're going to tell you about today. So this is an illustration of the type of problem we're talking about. This is a survey of over 1,800 clones assessed in Ubiaja, Nigeria. Now, Ubiaja is the very best location for getting good flowering in Nigeria. And it is the location that the IITA Institute is using as their location for doing their crosses. Nevertheless, in Ubiaja, in this set of 1,800 lines, we look at the flowering ability going from 0, which means no flowering at all, up to 3, which is good flowering. Only around 33% or so were good flowering. The other 2/3 were either medium or low flowering. In fact, 15 or 18% were no flowering at all. So what that means is that a lot of the favorable alleles that you might wish to use in order to improve the cassava are locked up in lines that you really have a lot of difficulty crossing. And so by finding ways to obtain better flowering, we have the potential of unlocking those that germplasm that could be used. So the problem is that cassava is late flowering. It takes a long time to induce flowering, maybe 4 to 9 months. And some lines, not at all. They will not flower within the 1-year time frame. Flowering among parents that you want to cross is often not synchronous. So we're just at the mercy of whatever the flowering behavior is. And if they're non-synchronous, that creates a big obstacle to making the cross of interest. we have a low number of flowers produced. So there's one thing is the induction of flowering, the other is the quantity or number of flowers. The low percentage of those flowers that are produced are female flowers. Peter will tell you about that. And finally, there's a lot of abortion or arrested development of the flowers, the fruits, and the seeds. In fact, each flower will potentially produce only 3 seeds, and oftentimes one or all of those 3 seeds do not develop because of the arrested development. So the goal of our project is to develop a set of tools, and tools that we will define as Peter will describe the type of work that we've been doing, a set of tools that breeders can use conveniently in their nurseries to hasten flower induction and to increase prolificacy in their seedling nurseries and crossing blocks. And we've been really pleased with the very great amount of progress that we've been able to make in developing a set of tools. And we're now in the conclusion of phase 1, where we have developed tools, and we're going to just enter phase 2, where we put more of our emphasis on working with breeding programs to implement them in practice. So back to this slide here, I just shows you again the participants, and we have representatives of each of these institutions who work with us on the flowering program. We do work under controlled conditions like greenhouse and growth chamber, and then we depend on them primarily to do the field studies. In Cornell, Peter and I are going to be telling you about the story. Sarah Adiomo just finished a postdoc earlier this year, and there's a little bit of information we'll provide from her work. Xian Guan, you may remember, she finished a master's degree about 1 year ago. And Deborah Ade is a PhD student with me. Currently, she's located at IITA and working on the program as we speak. So I think that's the last of my slides, and this is where I pass the baton over to Peter.
That sound all right? Good. Well, thank you for giving me the opportunity to share some of the work that we've been doing. I find this project really exciting. I think it's going to be beneficial in the long run, and it's been a really fun 5 years of work. I wanted to start out by showing some pictures and just giving a little background on what cassava flowers are like, because I'm going to be showing you a lot of data and make sure that it's understandable. Cassava, this is a cassava inflorescence. It starts out producing a few female flowers. These are open female flowers. And usually about 10% of all of the flowers are female. Second come the males. These are unopened male flowers at this point. And they flower at a forking event. So the apical meristem grows, and then it branches. There's 2 fork branches, and at that point, they flower. I'll have a little demonstration. Here's our cassava field, and this is what some grow. They grow up, they flower, they fork, they grow up again a little bit further, and they flower again. We call this the first tier flowering, and then this is second tier flowering. Generally, you get better flower development on the second, third, maybe fourth tier, and then it starts to taper off. But you get very little development of flowers on the first tier. Sometimes they grow like this, though, and you get no flower development on any of the tiers. I say no development. It does grow at very, very small inflorescence, less than 1 centimeter, and then abscisses and falls off. So no open flowers, no possibility of pollination. Sometimes they also grow like this for a very long time and then fork. And other times they just keep growing and growing and growing, and you get no forking, no flower development. It's actually kind of interesting too, because this is a desired trait in some areas, which is like a breeder's nightmare, breeding for a trait that makes it really hard to breed. So that's something that if we can stimulate artificially a plant that grows like this to flower, but then it, in the field, in growers' fields, it will still be growing like this. That would be wonderful. So this variability is seen across genotypes and environments. So you might have one genotype that flowers very well, another genotype that flowers very poorly. you might have a genotype that flowers very well in one location, very poorly in another location. So this is where we really first got started, the environmental factors. And two environmental factors that are like well known to regulate flower and influence flower development is temperature and photoperiod in other crops. And these also interact with each other. So that's where we started. And now I'm going to get into some of this, our data. I'll explain this a little slower the first time. You'll see this chart many times because this is how we represent the age of flowering or percentage flowering in most of our work. So the x-axis here is age in days, and the y-axis is the percent forking. This does not mean that they produced a large inflorescence. It just means they got to the point of forking. And a lot of times we're happy with that at this point. And so we have 3 different temperatures. These were grown in growth chambers here at Cornell. And what we saw was at a very warm temperature, 34 C, only about 22% actually got to the point of forking. A more moderate 28, we saw 75% forking, and this is a very cool temperature for cassava. 22, we saw 96% fork. And this is at the first tier. Second tier, we see a very similar pattern. So it really indicates that temperature is critical to flowering or forking. We also saw that at the 2 lower temperatures, there was an increase in floral development. So this graph here is, again, the days after floral initiation, so after forking. And then this is the number of flowers that are present on the inflorescence over time. And so what we see is that at 28, you get some increase in floral development. And then at 22, you get quite a bit more floral development. And the 34 is not on this because those never even forked. So I can't show any data regarding the number of flowers that were developed. Another question you might have is, so are the plants just really unhealthy at warm temperatures? Because 34 is pretty warm. And no, actually cassava here, it grows very well at 34 vegetatively. And at 22, it is much shorter, but it's a healthy plant, and you can see the forking and flowering. The next thing we wanted to look at was photoperiod. So the way we do our photoperiod experiments is you have regular grow lights for maybe 10 hours, and then we do a dim light extension. We use that's not photosynthetic radiation so that they're more comparable. We used red lights, red LEDs, in fact, because look at this chart here. This is the spectrum of the red LED, and it is the max is about 640, which aligns better to the phytochrome, which is the pigment that absorbs red light and stimulates flowering. The white LEDs you see are shifted away from the spectrum that's more beneficial for flowering and for absorption by the phytochrome. So with long day extension, and this is at a moderately cool temperature, 25, we saw that long days stimulates earlier flowering. You see here that at 10-hour day, it's about 40%, and at a 14-hour day, we're up to about 80%. And this data is representing 4 different genotypes. Most of the work we do, we have 4 different genotypes. Some are late flowering or poor flowering, and some are early flowering, and some are in the middle. So this is the means of all of those combined. And then at warmer temperatures, so at 30 degrees Celsius, we see also a benefit of long day in terms of flowering, that flowering, the percentage of flowering goes up. But it's not as pronounced as at the cooler temperature, which goes along with the fact that the first experiments we did with temperature showing that temperature inhibits flowering. Then we also wanted to look at more interactions between photoperiod and temperature. So, and this was done as opposed to a short and long day. We wanted to look at more what field-grown plants would be, which would be 12 hours, and see if we, just by going from 12 up to 16, you're also seeing a benefit. Because this would be more what's going to happen in the field. And indeed, we did. So this is, there's cool temperature and warm temperature. At the warm temperature of 30, or 35 day, 30 night, we see no flower initiation at 12 hours. But with cool temperatures at 12 hours, we see some initiation of flowering. And cool temperature at long day, even better flowering. So now I'm going to step aside, and this is a little bit, this is some work that Sarah Ediamo spearheaded, looking at FT, because it ties into the photo period. FT is a relatively new discovery that is the FT gene is responsible for flower induction. And she identified 2 FT-like genes in cassava. The model is that the photoperiod stimulates, long day stimulates FT expression in the leaves. It's transported via the phloem to the apical meristem, or the flowering region, and stimulates flowering. And this is common in a lot of different crops. And we looked at the plants we were growing in the different photoperiods. And indeed, one of the FT genes, FT2, was expressed at a higher level under long days. So it's indicating that the FT is responsible, or partially it's responsible for the flowering, the improvement in flowering that we're seeing under long day, under photoperiod experiments. So in conclusion, for the photoperiod and temperature, we see that cooler temperatures hasten flowering, and warm reduce flowering. Long days also decrease the time to flowering, and high temperature will reduce or eliminate that long day effect. So if you have, even with the long day, if it's very hot, it will not flower. And the low wattage LEDs are effective for stimulating flowering. And this is important when we get into field work. We wanted to have more efficient lights for the field. The next thing we wanted to look at was plant growth regulators. And we don't have to study this in detail, but I put this up here because often I explain to people that we use a few compounds, and they say, well, how did you know that those particular compounds were going to work? And the fact of the matter is we had no idea. We took a shotgun approach and tested lots and lots of plant growth regulators. And we tested them here at Cornell, and then our colleagues in Africa also worked on them. And lots of different compounds, different combinations, different concentrations. And we found in the end that there was 2 that are very important to flowering. Cytokinins, and we're using the synthetic BA, and then ethylene. And I'll both show work with ethylene, with STS, which is silver thiosulfate, which blocks the ethylene receptor and renders the plant insensitive to ethylene. And then ethafon, which is a compound that's absorbed by the plant and quickly metabolized into ethylene. It reduces flowering. So, silver thiosulfate. Like I said, it blocks the ethylene receptor. And here's our water control. Zoom in for you. This is what I mean by basically no flower. It's less than a centimeter long. You can see it's already turning straw colored. It's subsiding. It's going to fall off in a couple days. And then with spray application of silver thiosulfate, we see this enormous inflorescence develop. So this is really great. And here's graphical representation of the data we saw. This is just one concentration. We tested a few different concentrations, and we saw that with increased concentration, increased benefit. So here, this is water control, and then going up to 1 millimolar, we got the best benefit. So that was great, but it didn't work very well in the field. So we started testing a lot of different ways to apply STS. we had brainstormed a lot of different why this wouldn't work in the field. Maybe the leaves developed a thicker waxy cuticle. Maybe the rain is just washing the STS off and it takes a long time to get absorbed. So we tried some different adjuvants. We tried rain sticker that helps the plant, basically glues your substance of choice onto the leaves. We increased the humidity of the leaves, trying to keep the stomata open by putting bags over the tops of the plants. Some of them worked a little bit, but what we really found works well is this application using petiole feeding. So the way this works is you can cut a leaf off, and then there's negative pressure inside the plant. And so if you quickly cut the leaf off and put it into a solution, it will take up that solution through the vascular bundles. STS is not red. This is just to explain. And I was using this as a tracer. And so it is absorbing the red liquid. And then you can see the vascular bundles if you cut off that petiole rather quickly. A couple hours, it will be absorbed. We also cut this plant up quite a bit more and saw that it mostly goes up in the plants to the apical region. So this is a pretty effective way of applying STS. And we, again, tested a few different concentrations and saw that with increased concentration up to a certain point, we see increased benefit in the floral development. Okay, so this one I need to slow down for a little bit because it's a little complicated, but it's really exciting. So what we wanted to make sure was that STS was having effect on ethylene, and it's not just some extraneous factor of silver being in the plant that is causing the flowering. So we have two different treatments over a course of time. So at first we started out by either treating plants with water control or with silver. And these flowers were already developed. So this is unlike what we were doing in the past. This is a high flowering genotype we used. The flowers were already there. We sprayed them with water or we sprayed them with STS. Then we waited a little while, and then of the water controls, half of them we sprayed with Ethylene and half of them we did not. And of the plants that we treated with STS, also half of them we sprayed with Ethylene and half of them we did not. And if you look at this, the graph over here, so we have plants not sprayed with STS, and they have a decent amount of flowers, and then not sprayed with Ethylene, so no difference. This is our water control. Plants sprayed with STS, and then without Ethylene. Again, there's no real difference. STS does not negatively influence the flowers or positive because these are already developed flowers. If we don't spray with STS, so water, and then we spray with Ethylene, it knocks all those flowers off. Over the course, this is about 5 days in between these two treatments. And then if we spray with STS and then we spray with Ethylene, the STS protects the plant from the Ethylene response and none of the flowers fall off. So this makes it pretty clear to us that the STS is affecting ethylene in the plant, and it's an ethylene response. So I also said we worked with cytokinin BA. Again, showing you a lot of these controls with no flowers, but and we saw that BA had a slight beneficial effect on flowering alone, but it was not as good as STS. So initially we weren't that excited about it, but we kept being intrigued. But then what we saw was that the combination of STS and BA, there's a synergistic effect, and we get these very large inflorescences develop. This is really exciting. We're better than the STS, so this looks really great for us. And here is a graphical representation. You can see that the water control, and then we have STS alone, BA alone, but then with the combination, multiplicative effect, and it's much greater than the sum of the two. So that was wonderful, but it gets better. You can't believe it, can you? So we go back to this picture of I showed you before. And what I'm going to point out to you is, like I said earlier, typical flowering, you get a few females here. You can see this is actually fruit developing, and then males up at the top. So if I zoom in, oh, I went ahead. What we see is that with STS alone, you get males on the top. This is a typical ratio. But with BA, it's all females. The entire inflorescence is females. So we have about 150 female flowers on this. For a breeder who's trying to get seed, this is potentially very, very useful, this shift. And here's some of the data that goes along with this, so we can see that we get a slight increase with BA and STS, but then with the two, almost 100% females. We wanted to look more into this, and when do we need to apply this BA to get this shift? And what we did here was we have control and then the STS with no BA, and then BA 14 days before any visible flowering, at flowering, and then 14 days after. And what we see is you really have to get that application of BA on before you can even see any flowering. So it indicates that the transition to females happens very early in floral development. But you actually do see a benefit in floral development, applying even at flowering or a little bit after flowering, but you don't get the massive shift to mostly females. So in conclusion, for our plant growth regulators, we see that foliar applications of STS can increase the quantity of flowers. STS protects the flower from ethylene. Applying through the petiole is effective. The combination of BA and STS is much more effective, and that BA increases the percentage of female flowers. One of the last topics I'm going to touch on is pruning. So pruning, people have done this a lot, where you can manipulate source-sink relationships in plants by removing parts, by cutting them off. Our colleague in Colombia, Hernan Ceballos, started doing a little work on this, and he mentioned it to us, and we carried on with it also. And what we do is when the plant is very young, so earlier than this stage, and these lateral fork branches are less than a half a centimeter long, so very small. There's 3 branches usually, and then the inflorescence is also less than a half a centimeter long. You cut those branches off. So you get in there with a magnifying glass and a scalpel and pop them off, and then you get a nice inflorescence develops. This was really cool. So no plant growth regulators needed, and you get a huge inflorescence at the first tier, which the first tier normally doesn't produce many flowers, even in a high flowering genotype. And so this can really push the time to get flowers and to get seed quite a bit faster in the breeding nursery. We decided to take it one step further and see if BA would have the same feminization effect as we saw with the STS, and it sure does. You get about 100% females. So here's the data showing that you get an increase with both pruning and pruning in BA. So the next phase, as Tim mentioned, is going to be, we've kind of developed a toolbox and trying to disseminate this information out to breeders. We've been doing some trips and trying to develop red light setups so we can stimulate, basically mimic long days in the field. Using LEDs is really nice because they're low energy, and these will probably be set up on solar systems, solar powered, so they can be put in any field. Working with people like Simon Peter here to do some PGR applications in the field, and working with the folks like Smith, who are actually, he leads all the crossing at IATA for the most part. So not so much with the breeders, but with the people that are actually doing the work, doing the crossing. How can they best utilize this information and benefit their crossing the best? So in conclusion, just a few things. We saw that long day is beneficial. Cool temperatures around 25 seem to be the best. That's kind of difficult to manipulate in the field. STS and BA are great for improving flowering, and pruning also works. Some of the challenges are the STS in the field, we haven't seen as good of benefit as we have seen in the greenhouse. So trying to work on that. And then for phase 2, really optimizing these methods for the field, developing the best applications for them, assessing fruit and seed set in the field as they're doing the pollinations, making sure these methods work with crossing, developing field scale photo period extension, field testing of the pruning, best combinations, because there's a lot of different combinations you can do. Probably doing some of the photo period extension with the PGRs will have, I'm guessing that's where we'll probably end up going. And doing workshops training people how to use this technology. So thank you all for paying attention, and I find this really exciting. Hope you do too. And I'd like to thank our partners and our donors, because without them, wouldn't be possible. So I guess. Tim and I could field any questions.
I agree, it's very exciting, even to a soil scientist. So, so my question is, and maybe I kind of misunderstand the procedure, but Tim initially talked about using phenotyping of a training set to kind of predict certain properties and related to genotypic information, and then later on you're manipulating the phenotypical features. Is that an issue in terms of the broader program, or do you do the phenotyping much earlier on?
The phenotyping on the training set is a set of clones representative of the breeding population at large. And it's done without making any crosses. The breeding program requires the crossing in order to get recombination and favorable alleles brought in together. So that is done on this right-hand half of that diagram, where you would wish to do that cycling as quickly as possible. And the genotypics, GS, genotypic selection, allows you to have a model that's developed to relate the performance, let's say the yield, and the markers that are marking and sampling the genome, the sequence at single nucleotides positions with polymorphisms. You'd be able to model that, and then from the progeny that are produced at each turn of the cycle, you're going to do several thousand lines, analyze them by DNA analysis, and then from that DNA information, you will be able to predict what their breeding values are, whether they would be good parents to select in the next round of crossing. So what we're needed for is to help take young plants and get them to flower as quickly as possible, and in numbers of flowers as many as possible, so that when they have selected desirable parents, they can make the desired crosses on a timely schedule. And in fact, in some cases, there are lines that they would like to use, but they don't flower at all under conventional conditions. I don't know. I hope that helps you.
Yeah. Okay. No, I understand that now. It's just in different phases of the whole program.
We get the question a lot. Well, aren't you going to harm the farmer's yield potential? No, we're only working in the nursery with these tools. This is a breeder's toolbox. The farmers will not have an altered flowering plant if all goes, unless the breeding selection resulted in. In fact, in our case, it's likely that we would be selecting for low flowering lines more so than under conventional conditions. So it won't affect what the farmer will have as a genotype that they use. Dominic?
Yeah, I'm also thinking about the modeling. Have you got to a stage yet where you were able to assess how effective the model is, and when you've actually managed to freelance some of these and see how well the genotype is able to actually affect? Because that's the premise of the whole thing.
Right. So I'll take that question. And the question is whether there has been enough information collected to evaluate whether the process of genomic selection and the model development is effective. We did not talk about that part of the story. And in fact, there have been a series of seminars, primarily in the plant breeding seminar program, where Jean-Luc and Chidozi, Agise, and others, and several students, PhD students, have gone through the program and have also presented. But the answer to your question is that yes, there's evidence that it is working. Is it twofold, tenfold? No, it's not that kind of thing. It's a quantitative trait. Breeding for yield, which is one of the primary targets, is a process that is going to result in steady but relatively small improvements over time. You're talking 1 to 10%, 1 to 5% increase per cycle, let's say. So it is working, and so we're encouraged to go to this phase 2, which we're just entering now.
So, what very interesting results. So what I wonder, so you have such a big variation in terms of flowering between different cassava lines. So your data imply that perhaps this variation is due to endogenous ethylene level, like, you know, high flowering lines have low ethylene and high cytokine. So have you measured, have you done that with any analysis?
You should take that one, because you showed the data.
I think you're asking, have we done the analysis to show that in a plant that we're not treating, does ethylene?
No. What I wonder, so in the first slides, when Tim was showing that like there are 33%, only 33% of cassava lines have high flowering potential, but like there's quite a bit of variation in terms of how much flowers are produced. So what I wonder, whether this variation between lines can be also due to this inherent ability of this line to produce ethylene. For example, those lines which consistently produce lots of flowers, they actually produce less ethylene. Like there's like a natural variation between lines.
Yes, that's a very good point. And we haven't measured ethylene production across genotypes at this point, but it's perceivable, or could be that.
What little we have done, and this was when Xian Guan was a master's student, she finished a year ago, and she tried a few different lines. There wasn't that particular factor, the ethylene, was not the key differentiating property. So I'd say that's not terribly surprising, because you have really in the process of signaling for both flower induction and for flower set, quite a lot of different things that could be genetically different between lines. And so that, at least in the lines that she surveyed, very small, 419 and 0002, that wasn't the key thing. Similarly, when we look at the FT production, while the genotypic, the photoperiod difference in the FT2 transcript levels was showing very clear relationship to photoperiod. If we look at the genotype differences, that transcript abundance alone wasn't a good predictor. So clearly there's many other components of a signaling pathway that could be different between lines that could be responsible for the overall outcome. It hasn't been our objective to do a complete and thorough investigation of what causes these things. We've been very much in the applied science mode, where we're looking for this toolbox, things that will work for the breeders. Sadly, as a physiologist, we have not been given the responsibility to pursue molecular physiology in detail.
With the, so when you produce the next generation, you've made your selection process. So you're speeding up that cycle. When it goes to the grower, are you then cloning, taking clones of the selections that you made?
That is correct. Yeah. So it is a clonally propagated crop, and once it's released as a clone, then it will stay the same as long as there hasn't been any. Well, it should stay the same over the years.
So is that a limitation? That makes sense.
It has pros and cons. I mean, you do fix the genotype by the cloning because it's a clonally propagated crop. That can be an advantage. Once you have created something that's valuable, it stays, it can be propagated and kept the same over many, many years and in many locations. It does have the obstacles in the breeding process that are what I would consider challenges.
But I thought you were actually producing clones as unique. That doesn't sense like
well, yes, but that's part of the conventional agriculture in a clonally propagated crop. In the case of cassava, what it entails, there is this issue of once a breeding program has developed something superior and they're ready to release it, then there needs to be a propagation process. At IITA, they've been experimenting. They have a nice method for doing this in a kind of semi-tissue culture. It's more explant culture under aseptic conditions. And then when I was there, for example, they had thousands of little boxes, sort of plexiglass. They would ship it out to Catholic Charities, and then Catholic Charities in turn would plant them out where the plants would be grown for one cycle. And you get a process going where you can quickly amplify or propagate to large scale for a release. Similar things have to happen in any breeding program where you go from initial small little handful of seed to you want to have now thousands of bushels. If you read the story about Norman Borlaug releasing lines in India, you know, that's a really big part of the whole process. And in cassava, it's especially challenging because it is this vegetative propagation, but it is it's doable, and it just requires some have some resources, let's say, devoted to that. Yeah. But there is the advantage that once it's released, then it can be shared with neighbors, and it's always identical, and it can be disseminated and spread if it's an acceptable and highly desirable by the community.
So just a real quick question. You mentioned in your kind of future vision that you were planning on saying if there was an effect with the red light with PGR together. Have you done any initial studies on that, and is it synergistic in addition to what you're getting with the STS plus VA?
We've done some small studies and shown that I wouldn't say it's synergistic, but they do work together well, especially with what you said, the red light and the like the STS. Those are kind of two different factors because one is dealing with the timing of flowering and one is dealing with flowering abundance. And we did want to make sure that, you know, it still works under the red lights, and it does, but we didn't identify really a synergistic effect.
That is the sort of thing that, for example, Hernan Ceballos at CIAT is actively working on, as well as our colleagues at IITA and particularly NRCRI in Nigeria. We'd like to see that kind of work done, sort of taken up by our breeding programs at this point.
Thanks, everybody. Thanks for coming. And
This has been a production of Cornell University, on the web at cornell.edu.